Today we will discuss about the history of microbiology. As we all know that the microbes are an indispensable component of our ecosystem. Indeed, modern biotechnology rests upon the microbiological foundations. Mankind have known the use of microorganisms since very long before the proof of the existence or the discovery. Father of microbiology is Antony van Leeuwenhoek. He was also known as the first microbiologist. In history, the golden age of microbiology or the golden period of microbiology is described from 1860 to 1910. Certain landmark accomplishments that were performed during the golden age of microbiology are as follows. We will discuss certain scientists which provided or which proved the discovery of certain things. First is the Robert Koch. Robert Koch proved the germ theory of diseases. He also isolated the bacteria causing anthrax and tuberculosis from the infected humans. Is the Louis Pasteur. Louis Pasteur disproved the spontaneous generation theory. He was responsible for the discoveries of rabies vaccine, and he was also responsible for the discovery of certain immunization techniques. Third is the Christian Germ. Christian Germ was known for the development of Gram staining technique. Fourth is the Joseph Lister. He was responsible for the discovery of certain antibiotic methods in the surgery. And fifth is the Robert Hooke, that is, he provided the description of cells in the piece of cork. Now let us discuss the discovery of microorganisms. In order to prove the existence of microorganisms, certain theories were put forward to predict that microorganisms are present in the environment. The certain theories were the theories of spontaneous generation or abiogenesis, second is the theory of biogenesis, and third is the germ theory of diseases. Now let us discuss about each one of those in detail. First is the theory of spontaneous generation. In earlier times, people believed that the organisms can develop spontaneously. That means an organism, a living organism, can originate from a non-living thing as well. But Francisco Redi performed certain series of experiments which proved that the organisms generate spontaneously. He took 3 flasks containing piece of meat. In each of the In case of first flask, he did not cover the flask, and after a certain period of time, maggots appeared. In the second flask, he covered the flask with a piece of paper and observed that there were no life forms appeared. And in the third flask, he took the flask and kept the piece of meat inside it and covered the flask with a piece of gauze. That means it was in the form of a sieve from which the air can thoroughly pass in and out. After a certain period of times, we saw that the flies were attracted towards this flask and maggots were appearing. This was about the spontaneous generation theory. Now let's discuss the theory of biogenesis. In order to disprove the spontaneous generation theory, Louis Pasteur suggested that the dust carried the germs, and if no dust was present in the environment, the broth or the boiling of the meat can remain sterile for indefinite period of time. He also believed that the spread of organisms can be controlled, so the diseases can also be controlled. He performed a series of experiments to prove the biogenesis. He also took 3 flasks for the experiment. In all the 3 flasks, he took sterile broth, the first flask was opened to air, and after some time we saw that the life forms appeared in the first flask. In the second flask, the neck of the second flask was sealed, and we saw that no life forms were appeared in the second flask. In the third flask, what Pasteur did was he covered the neck of the flask and he opened a tube-like structure which was heated and was opened to air, and we saw that when it was opened yet heated to air, we saw that no life forms were observed, which proved that the organism do not generate spontaneously, whereas it needs certain living parent organism to develop a new organism. By proving this theory, Louis Pasteur disproved the theory of spontaneous generation and told that organisms always require a parent organism to grow. Third is the germ theory of diseases. Germ theory of diseases was proposed by Robert Koch, and Robert Koch presented his theory and gave the final proofs that bacteria can be isolated and cause diseases. He made series of experiments in the lab and led the establishment of Koch postulates, which provided a guideline for the identification of organisms. The Koch postulates are: a specific organism is always found in association with some diseases. Second is the specific organism can be isolated and regrown in the laboratory. Third, the pure culture of the same organism will cause the same diseases when inoculated in the susceptible host. And last, it is possible to recover the same organism from the pure culture from the infected animals which were performed experimentally. Now let's discuss about the fermentation. At last, in the discovery of microorganism, it was proved that an organism always requires a parent organism for their growth, and it is possible for the organism to be regrown inside the laboratory in the in vitro conditions. Now, Louis Pasteur suggested that the process of fermentation fermentation of fruits and certain grains are carried out by the microbes. He also suggested that undesirable microbes can be avoided by heating the juices, and the heat should be not enough to destroy the flavor of the juice, but it should be enough to kill the unwanted organisms that are present inside the juices. He found that heating the juices at 62.8 degrees Celsius, that is 145 degree Fahrenheit, was doing the job for half an hour, and this process is known as pasteurization in modern times. And these pasteurizing techniques is performed or is carried out on a daily basis in certain industrial areas like the dairy industries. That's all for today's session. Thank you.